Welcome everybody to today's webinar, Advancing AAV Vector Production with High Integrity DNA Constructs, and thank you for joining. My name is Dr. Marcello Stein, and I'm the Marketing Director at PlasmidFactory and will moderate this session. Let's start with some technical introduction. This webinar will be recorded and made available to the registrants. Participants are muted, but can pose questions in the chat using the question mark symbol in the upper right. Questions will be only visible to the moderator and read out at the end for answering. Please adjust your viewing window in the center according for best visibility using the zoom buttons. So now it is my pleasure to introduce our speaker, Dr. Shankar. Dr. Shankar is Chief Scientific Officer at PlasmidFactory and joined the company in 2014. He did his PhD in biotechnology at the University of Bielefeld and now leads the R&D department with his team, overseeing plasmid and minicircle development in a variety of application fields. So, Dr. Shankar, the stage is yours.
Right. Thank you, Dr. Stein. Thank you for that introduction. And hello to everyone. Welcome to this PlasmidFactory webinar on advancing AAV vector production with high integrity DNA constructs. The agenda in this webinar would be a very brief introduction to our company PlasmidFactory, and then I will go straight into the different quality grades that we offer. And then we will look into our special flagship product, that is the Minicircle. And then we will talk about different solutions for AAV vector production tailored to your needs. Okay. We will be looking at the two-plasmid system as an alternative. We will be looking at how minicircles can change your AAV vector production. And finally, let us get into the important topic that has recently come up, the AAV inverted terminal repeats and what they can do to impact your vector production. All right. This is the history of our company. In exactly about 3 weeks from now, we will be celebrating our 25th year of existence. So we've been there even when the field was in its relative stages of infancy. And we've been seeing the field grow and mature. You can see those sparsely populated points during the 2000s, relative quiet years, but we were quietly building up our capabilities, our competencies, acquiring technologies, working on them, perfecting them, filing important IP applications. And in fact, you can see those important milestones in 2011 and 13 where we achieved patent applications for the production of AAV using minicircles, which we'll be looking at in detail in the further slides, and also application of minicircles for CAR-T cell production, which has seen tremendous success in clinical trials in Germany as well as in other European countries for adoptive immunotherapy. All right. And then when the pandemic hit, we were there to deliver, where we produced high quality grade plasmid DNA in large quantities, which acted as starting materials for testing and producing mRNA vaccine candidates. And then in the recent years, you all know how the gene therapy field has developed and matured. So you see so many approvals and concomitant development in our company as well. We have expanded our production capacity. We have a new investor which are backing us now. And we are now poised to enter the next logical step, that is GMP production. And we are excited to go along with our customers and supply their needs as their needs change and as the field matures as well. So this is a nice snapshot of our company, and personally for me, it also in some way reflects the development of this field in the past 25 years. So our portfolio is basically custom plasmid DNA production for our customers. So when they have started already producing small amounts of their candidate plasmids, which have tested, and they want to move into preclinical studies, they then ask a CDMO to produce them. That's where we come in, producing your plasmid DNA in a higher quantity and in a defined quality. And of course, we also produce minicircle DNA, which are specified to your needs and your gene of interest and so on. And apart from this, we have in-stock DNA with some reporter genes, but the most important one, the relevant one for us in this talk would be those in-stock DNA molecules that are going to help you accelerate your AAV vector production. So plasmids and minicircles that are tailor-made for AAV. Okay. We have different quality grades. We have starting from our scientific quality grade, which is the best suited one to just start off and produce some plasmid and test them in your preclinical studies. And this is already starting from about 5 milligrams for research grade plasmid DNA. But then you can go on to high quality grade once you've identified a candidate. And then these are actually already sufficient for you to allow for you to enter GMP production of your viral vectors for early clinical phase studies. Okay? And then you have your GMP grade, of course, where everything connected with late clinical phase of market supply and so on, and of course, application of DNA directly into a human patient. So, all right, so I spoke about our solutions for your AAV vector production. We are going to start off with our two-plasmid system. So you have your conventional three-plasmid transient transfection, which works great, no doubt about that. But when you think about trying to produce your viral vectors in a large quantity, and when you try to scale up and actually go into commercial production, you need to think about all factors that will help you along the way to kind of ease your way of producing your viral vectors. And switching to a two-plasmid system is one such method of easing your production. Okay, so we have your transfer plasmid containing ITRs, and the adenoviral helper as well as your AAV packaging functions are all packed into the second plasmid. So you do not compromise on safety while still maintaining the vector yields that you have previously seen. That is possible. So what we do offer here is that a helper and packaging plasmid that is corresponding to the AAV serotype that you're going to work with. So the cap genes will change while all other genes will remain similar. And then you have your custom-made vector plasmid with the ITR flank transgene. So you can start off with a reporter gene that is in stock, and you can test your systems and optimize and find out the right conditions, and then go for your transfer plasmid. What this will result in is a simplified AAV workflow. You have helper and packaging on one plasmid, so fewer components. So the complexity of your transient transfection is essentially reduced. You don't have to play with too many variables to try to hit your producer cells with the right components. So you kind of reduce the complexity on that front. And you have consistent transfection performance. This was shown in studies that we are going to show in a few minutes now, and with a relatively lower DNA input. Okay? And what this then, as I said earlier, what this data means is that when you produce these plasmids in a GMP ready stage, then you kind of ease your production of viral vectors because you reduce your costs, less amount of DNA to be produced, less QC burden, and so on. So essentially, you reduce your gene therapy vector price. We have also successfully integrated these constructs with our Minicircle and our ITR Protect technologies. We'll be looking at it further down. So this is nicely integrated. You can look at the two-plasmid system as a solution that can be readily applied if you are already in a three-plasmid system. So this was done by our collaborator Rythera, where they tested a transfer plasmid 1451 here, coding for a GFP reporter gene. This is for a single-strand AAV, and helper and packaging plasmid 2432. This is for serotype 2 AAV2. So transient transfection, your recombinant AAV particles, they tested different ratios of these two constructs and found out one ratio to be particularly effective. They tested the different total amounts of DNA and the best transfection reagent. And the results that they achieved was interesting. If you see the shake flask cultivation, you have about 10 to the 10 vector genomes per milliliter, which is actually quite in the range of what you would expect in the field. So this is in no way a compromise in the vector yields that you would see. And importantly, they were successful in transferring this into the bioreactor cultivation. You see the amount in the vector genome yield actually increased a bit twice. So this might still be within the limits of variation, but it is important to know that this was consistent, it was maintained, and it was actually slightly increased. Getting 10 to the 11 vector genome per milliliter is actually pretty good value. And importantly, they also had 10 to the 8 particles per milliliter transducing titers, which is an important factor to consider when you are looking at your application. They were also successful in working on their own proprietary cell line. This is the rice cell AAV, which was able to use this two-plasmid system and produce vector titers of 10 to the 10 vector genome per milliliter. And importantly, the infectious titer in this case was even slightly higher than what they saw earlier with the commercial cell line. So these examples actually show that it is possible to take the system and test it in your own conditions and apply them and successfully transfer from a 3 to a 2 plasmid system in suspension culture. All right, and then we also had a collaboration with Sartorius. This was during the time when we were testing different systems for single-use fermentation, because we were in the stage of preparing for our GMP productions, and it was very clear that we had to transfer completely to a single-use environment in upstream as well as in downstream. When it comes to fermenters, it is usually the belief for a long time in the field that these single-use fermenters, they are well adapted, well suited for animal cell culture, but with microbial cultivations, it is quite doubtful. And we started these experiments, and we were pleasantly surprised by their STR microbial single-use cultivation system. The performance of these systems, these bags, in the amount of biomass that it generated and also in the amount of DNA that we could isolate, we could not find a difference between the levels that we were achieving with our conventional stainless steel fermenters. So this was an important milestone for us to actually be able to transfer our steps to a GMP environment. And to show the similarity in the quality of the DNA, I've shown you two gel pictures here. On the left, you have DNA for AAV transfer plasmid. This is a GFP reporter gene for single-strand AAV. And you see SS, that is your conventional stainless steel fermenter. And on the right, you have your single-use fermenter. So the DNA that was isolated from two different systems, they were practically similar. They were not possible to differentiate between the qualities of these two DNA. And also the productivity was similar. And on the right, you have the example of a helper and packaging plasmid. These are these two big 22 kb plasmids. So you have essentially the same level of quality using your single-use fermenters. So what we also see here is that we are able to achieve a very good AAV genomic titer as well. They took these DNA and tested them in their transfection protocols, produced AAV vectors. This is in fact for AAV6. And they found out that they were able to achieve 10 to the 12 vector genomes per liter. I understand this is a little bit lower than what you would expect, but still it is a pretty reasonable vector genome titer considering that a different transfection reagent was used, which is not the most optimal one for AAV vector production, but still being able to achieve 10 to the 12 vector genomes per liter is actually a pretty good value. So what it again shows is that the application of the two-plasmid system and its proof that it works in suspension culture is now a given. Okay, now I will come into the concept of Minicircle. So we basically take your gene of interest, your sequence, in this case, your ITR flanked sequence, along with the ITRs, we can clone it into our production system. And then once we amplify that plasmid, even within the cells, we trigger a reaction to remove the bacterial backbone specifically out of that molecule, while leaving behind a supercoiled monomeric molecule called the Minicircle. Now, I need to stress on this point that it is important to know that the molecule that is left behind, that is Minicircle, comprising of almost only your gene of interest sequence, is still supercoiled. Okay, we can actually test them with quality controls that you would normally apply for a plasmid. So on the left, you see an electropherogram from capillary gel electrophoresis. That large peak that you see, that corresponds to supercoiled DNA. Okay, only that this is actually from a finished purified Minicircle DNA product showing 99.8% supercoiled DNA conformation. Okay, on the right, you see an agarose gel, you see the supercoiled Minicircle. You can linearize it, you can find that the band shifts upwards. So you can do all those quality control tests that you were doing for plasmid DNA, and you can still achieve the same reliable results. So being a supercoiled molecule, but now that one that contains only your gene of interest, is an interesting phenomenon because these molecules do not have any resistance genes, they don't have any bacterial markers. So essentially they mean they bring less immunogenicity into the picture. So later on, when you produce your vector particles and you apply them into your target cells, you have a less likelihood of eliciting an immune response. Okay, and they also have a higher transfection efficiency while producing your vector particles. Thanks to their smaller size, they have a higher chance of entering the nucleus. And more importantly, the particles that you produce are not just going to be less immunogenic, they are also going to be protected from epigenetic silencing. Because simply the absence of bacterial sequences are going to make sure that the particles that you produce and the DNA package that you deliver into your target cells is having a less chance of being shut down due to being associated with bacterial sequences. All right. The most important advantages relevant for your applications are the ones that I have highlighted right down in the bottom. This means improved yield of your AAV vector production, which will minimize your costs, of course, and no back packaging of bacterial sequences during your vector production, which will increase tremendously the purity of your vector particles. So we're going to look at this in detail, and we are going to see how our tailored AAV solutions will help you to achieve these two objectives. All right. So we saw first how we can shift from a 3-plasmid to a 2-plasmid system. So what if we could take just one of these molecules from a 2-plasmid system and convert them into a minicircle? So in this case, just the transfer construct has been converted into its minicircle counterpart, flanked by the ITRs, whereas the helper and packaging construct remains as a plasmid. This was a work by our collaborator, Excel, which is now belonging to Sartorius. It was very interesting where they tested 4 different media, and in each media, they tested plasmid as well as minicircle components, okay? And in the plasmid combination, they had a transfer plasmid from PlasmidFactory for single-strand AAV, as well as the helper and packaging plasmid. So this was for AAV2. And in the minicircle combination, they just replaced the transfer plasmid with the minicircle DNA while keeping the helper and packaging as a plasmid. Just by this change and by keeping the molar ratio similar in both cases, they were able to see that in each media, the amount of vector genome titer achieved with the minicircle combination was always higher than when using plasmids. Importantly, this increase was not due to a result of more cells being alive and producing more particles. You can see those gray squares, they represent the viable cell density, and they are similar in both cases for each media. So this increase in vector genome titer is actually coming from more particles being actively produced per cell. Okay, this is an important observation. And if you look at their own in-house medium, which has been optimized for AAV vector production, the third medium, the Excel in-house, you see that the best increase was seen, and this was as high as 4-fold higher vector genome titers compared to using plasmids. All right, so we saw what we could do with converting just one construct into a minicircle. So what if we converted both the constructs into minicircles? So this is where we have a transfer minicircle containing ITRs and a helper and packaging minicircle. Now we enter the realm of vector purity. Okay, so this was another collaboration with our Hildegard Büning's group in University Hospital in Cologne, where they first tested our 2-plasmid system. You see the vector construct as well as the helper construct. And what you see here is a combination of vector and helper as plasmids, and you detect backbone sequences in your vector particles. Okay, these the target was here the ampicillin resistance gene, and these are detected at about 10 to the 10 here, which actually corresponds to about 2 to 3 or 4% of number of vector particles totally produced. Okay, this is actually quite high when you consider the number of vector particles that one would later inject into a patient when you're in a clinical trial. So this is actually quite a high number. And just by converting the vector construct into a minicircle, you see a significant drop already. And by converting both the constructs into their minicircle counterparts, we went right below the level of quantification. So this was a tremendous increase in genomic purity, which does not get appreciated currently, but once we reach that stage of regulatory compliance, we need to look at these aspects. And these questions have already been put forth by the FDA in their guidelines and so on. We see these things now coming up, and it is important for our customers and clients to know what options are there to increase the vector genome purity, because this is really becoming an important case now. In case of self-complementary AAV, one has to note that they actually found also an increased transduction efficiency. This was as high as 30-fold while using self-complementary AAV. So you could keep your vector genome titers because you're producing the same, because you're giving the same number of molecules on a molar basis. So you can keep your vector genome titer, but you can tremendously increase your vector genome purity. Okay. All right, so we move on to the inverted terminal repeats. So this is latest research by our academic collaborators at the Bielefeld University, Christian Müller and Marko Radukic, who have done some tremendous work in this field in the recent past, and shown that it is important to look into your actual ITR sequence and the status of your ITR sequence within your plasmids. This has been neglected for way too long, and it makes sense to look into it, and that is what we'll be seeing in this slide and the coming slides. So on the left, you see your typical AAV vector genome representation. We all know this, the single-strand AAV vector genome with the double-stranded hairpin ends, a complex palindromic repeat that you find there. But on the plasmid, this is how it will look like. On a double-stranded plasmid, you will find the sequence represented like this. And what they achieved was to clone the actual full wild type length ITR sequence from AAV into a plasmid and have their transgene in between them, and found out very important differences to a typical plasmid. Okay, so what you see here are 3 different parameters to test the production of your AAV vector. And in each parameter, they tested 3 different combinations. You see 2 ITRs at the 2 positions. You have a 130 base pair, which is a truncated ITR, and a 119 base pair, which is a truncated plus internally deleted ITR. So this is one combination. And in another combination, they used 2 truncated ITRs, that is 130 and 130 base pairs. And in the third combination, which is their newly cloned versions of full-length AAV ITR sequences as they appear in the wild-type AAV virus. Okay. They found out that the third combination is giving a significant increase in the yield of AAV vectors produced per cell. So this is important to note that this is yield per cell. So this is not a volumetric effect or so on. This is actually more vector being produced in every producer cell. Okay. So this is actually very interesting because this is important, because when you look at gene therapy vector production, you need to look at every single point where you can increase a little bit more of your vectors. So this is important. Whereas in the second picture, you see genomic purity. So this is measured in terms of the number of backbone sequences that you actually detect. Okay. In the case of your typical plasmid that you would usually find in your AAV transfer plasmid, the one with truncations and deletions, you measure up to about 2.5% of backbone sequence containing particles. This is actually quite high, as I already told you. But just by converting both of them into their truncated versions, you see a significant drop. But once again, when you look at the full-length ITR sequences, these plasmids result in vector particles containing just about 0.5%. And this is actually a very significant reduction, although it might not sound so. A reduction from 2.5 to 0.5% is actually very significant. Okay. And for me, the most interesting aspect here is that these are plasmids. So these sequences are actually present in the starting molecules that you give into your cell, but the cell still selectively only packages the transgene. They don't package these backbone sequences, and that is the effect of having these actual wild type length ITR sequences. And further to the right, you see the packaging efficiency. So this is not a significant increase, but still you see a very clear trend of higher packaging efficiency while using wild type length ITR sequences. So this makes it very clear and this actually represents the motivation of why people should start taking a hard look at their ITR sequences in their transfer plasmids, because this has tremendous room for improvement. So we are going to bring this all together into our ITR Protect, ITR Rescue portfolio. This is how we commercially plan to offer it to our customers. So you come to us with your transfer plasmids, we look at it, we can characterize them with NGS sequencing, we can convert them to the full-length ITR sequence to help you achieve the actual potential of what you could do with your transfer plasmids. And then we produce them, and more importantly, when we produce these plasmids, it is important to follow our procedures that we produce them according to our modified fermentation processes, which will help you stably maintain these sequences in their fully cloned full-length ITR level. And we can monitor them, we can quality control them, and what you get is then a product that has a higher homogeneous level of ITR sequence. So to show you an example, this was an experiment that we conducted in our labs to actually show how ITR degradation can take place and how we can control them. So within 10 generations of cultivation, you can see that ITR sequences can get deleted. Okay, the red points are one of the ITRs in the plasmid, and the gray points are another ITR. This is a proximal one to the one proximal to the origin of replication. You see there is this high peak. This is the one. This is the level of the full-length ITR. But within 10 generations, you actually see another peak coming up. So all of these points correspond to those plasmid molecules that have started losing a portion of their ITRs. This actually is true data. This is possible. This is what can be expected when you normally follow your fermentation conditions. So this was not known for a long time, but now we have those tools in our hand. We have NGS, we have the possibility to really look deep into our plasmid sequences and say, okay, what is the actual status of my different plasmid molecules within this preparation? And therefore, when we follow optimized fermentation procedures, according to our ITR Protect technology, we can effectively suppress the rise of these truncated variants. You can see here that this peak has disappeared here. And while applying the full range of possibilities or points that we have within the ITR Protect portfolio, we can now achieve, you see on the right, extreme right here, this ITR condition, a plasmid with such high levels of ITR sequence homogeneity that has not been seen before. And this is possible, and this is what we offer now within our new ITR Protect technology. What this actually means for you is that when you look at your plasmids, you can follow your new optimized fermentation. What we follow is the new optimized fermentation conditions, which result in consistent. Here we look at different clones after a typical transformation. We can actually expect a consistent performance of these clones in maintaining these very fragile full-length ITR sequences in the condition that they were cloned. Okay, an already deleted ITR might not undergo deletion that quickly, but when you want to see those benefits that you saw earlier in those slides, then you need to have your ITRs in their full-length condition, and they are more prone for new deletions. And so it is important to follow these optimized conditions to maintain them at that level, and that is what you see here. And this is about 89, 88%. And it is important that you don't find 100% here, because then you will have to start suspecting the data. This is actually not possible to reach 100%. But what you see here is a consistently high level of maintenance of your correct ITR percentage. Whereas on the right, when you follow your normal typical standard plasmid production procedures, which is actually pretty okay, but then you can see that you do not have the consistency. You will start losing ITR length faster than what you would expect in your optimized fermentation conditions. Okay. All right, so we spoke a lot about mini circles in the beginning and how they would help increase your vector production. And then we spoke about how full-length ITR sequences are important. So we had this idea, why not combine both? Is it possible to make a mini circle with full-length ITR sequences? And if yes, what can it do? This is what we did recently. This was very interesting for us to see that we can actually produce these mini circles while maintaining the ITR sequences in their full level. So these two processes are not exactly similar, and we were successful in bringing them together compatible that we were able to produce a minicircle here in this case for a GFP reporter gene, and we can now produce these minicircles as well containing full-length ITR sequences. And when our collaborators, Marko Radukic, Christian Müller, when they applied them for AAV vector production, we had some very interesting results this year. So on the left, I've just shown this small image that you saw earlier, 2-3 slides back. The best condition that you saw in this image, the one with the full ITR plasmids, how they increase the yield that you saw earlier. If you take this condition as normalized to 1, it is now possible to just replace the vector construct with a minicircle and then push up this genomic yield further 3 times higher. Okay, just by changing this. So this is the real combination of using minicircle and full-length ITR sequences. When you use full-length ITRs in plasmids, they resulted in very high vector genome titers. Now bring that down to level 1, normalize it. When you just convert the transfer plasmid to a minicircle, you can push it up 3 times even further. Okay. When you look at the parameter of purity, you see, of course, as I would expect, there is a significant reduction in the detection of falsely packaged backbone sequences. And then when all 3 constructs were converted into their minicircle counterparts, as we expected, this detection went below the level of quantification. So this represents absolutely pure vector genome particles that you would not otherwise be able to get. Again, looking at the packaging efficiency, you see a very clear increase in the fraction of vector genome to the capsid while using all the constructs in the minicircle conformation and while using full-length ITR sequences. To put it simply, we have higher titers, we have high purity due to low mispackaging, and we have a better capsid loading. Okay. And considering all of these together with our GMP facility, and we have our high quality production, you can actually start producing your GMP grade AAV vectors using these best conditions possible. So I'm going to summarize all of this information in this one slide here to just kind of take a look at what we saw so far. We have our typical transient transfection of for AAV, your 3-plasmid system, which works, no doubt about it. It's great, but we now have to start thinking about every possibility of easing your viral vector production when you move up the ladder and try to produce in a large scale. And one such possibility to help you probably ease your production is a 2-plasmid system, where the rep, cap, and helper genes, which do not have to be optimized further within themselves, they can be fixed into one construct. That will help you massively because this construct can be produced en masse, reduce costs, and then you have only one other plasmid. It will help you reduce your optimization costs. You need to only deal with 2 plasmids, and rather the whole production costs and the quality control costs can be reduced. Then we saw about how shifting from plasmids to minicircle DNA has a tremendous impact on both your vector genome yield as well as your vector genome purity, because of the fact that you remove the redundant and actually detrimental bacterial backbone sequences, you end up with clean, safe, and pure AAV vectors. And in the case of self-complementary AAV, the removal of these backbone sequences actually increase the transduction efficiency 30-fold. Finally, we looked at plasmids containing wild-type length ITRs, how they improved yield, purity, and packaging efficiency. We also saw how it was now possible to actually monitor and track the lengths of the ITR sequences in your plasmid population and stably maintain them during bacterial cultivation. Finally, the combination of these wild-type length ITR sequences and the PlasmidFactory minicircle technology was key in shooting up these parameters even further, increasing the yield 3-fold, increasing the genomic purity by bringing down the detection of backbone sequences below the level of quantification, and by increasing capsid loading. So this represents the latest in this field, and this is an unsurpassed technological advantage ready for you. All right, so what we have in stock is very important. This is off the shelf. That means you can get it very quickly and with a relatively lesser cost. We have a row of helper and packaging plasmids depending on the serotype that you need. This is from 1 through 6 and as well as AAV 8 and 9. And recently we were successful in converting all of these constructs into their Minicircle counterparts as well. On the right, you see some typical GFP reporter gene vector constructs. These are your ITR flanked transfer plasmids. You have your plasmids for single strand as well as self-complementary AAV as well as minicircles for both of these AAV conformations. And recently, we were successful in also producing in-stock wild-type length ITR containing plasmids as well as minicircles for single-strand AAV. And of course, apart from this, you may have your own customized cap-modified plasmid. Of course, we can produce them as a CDMO. You may have your own, of course, therapeutic gene containing ITR flanked transgene plasmid, which we will be happy to produce. Right, this is the team in proudly posing in front of our new GMP facility. If I have to summarize my whole talk, I would begin with those plasmid and minicircle constructs that are going to be key for maximizing your AAV vector production. That is the goal here. And we have different solutions tailored to your needs. As how your need might differ, we might have a solution in that area. I showed real and important application data from different collaborators. These are the proof that these technologies work and they are ready to be transferred to your system. We have been in the game for 25 years. We are a player with reliability that's backed by experience, and therefore we know usually what our customer needs. And those needs have matured and changed with the field, and that brings me to the last point. We are happy to go along with our customers to the next stage, and we are just before our GMP production. We are going to start in just about weeks time now, the first GMP production, and we are very excited about that. I thank you all for your interest at this moment, and yeah, I hand it over to Marcello.
So thank you very much, Dr. Shankar, for this very interesting talk. Really great. The slides as well as the recording will be shared soon with all the participants and registrants of this webinar. And just to make you aware, you can also download our AAV flyer in the Material tab on the upper right of your webinar window, and to get some further insight and a link to our webpage. Well, and we also received actually some questions in the chat. I will now read them out one by one for Dr. Shankar to answer. Let's start with this first one. You showed several data sets where switching from plasmids to minicircles increased AAV titers. Can you explain again how minicircles could actually be influencing viral yield?
Yeah, that's a good question. No, apart from the aspect that they might possibly be entering the nucleus a bit more effectively than compared to plasmids, thanks to a smaller size, what we think that might be going on behind the screens is that by removing those redundant sequences, we are probably directing the producer cells to a scenario where it is more likely that only the recombinant genome between the ITRs is getting amplified and packaged. In fact, there was a recent report that suggested that there might be some cryptic TRS-like sites in the plasmid backbone sequences. So now, thanks to NGS characterization, we have the possibility to look into the genetic structure of recombinant AAV particles like never before. We can look in depth. And I think such aberrant rescue sites might help explain why we observe certain backbone sequences in AAV vector particles, and we also sometimes observe backbone plus transgene chimeras. Okay, so basically, all these events will decrease the efficiency of your AAV vector production because they will compete for resources. And these aberrant molecules, if they are within the packaging limit, I would expect they will also end up in particles, so thus making capsids unavailable for the actual transgene genomes. So all of these factors might play together to see the observed increase in vector genome yield while using minicircles as compared to plasmids.
Great, thank you very much. Second question is a short one. What transfection reagents were used?
In one study, they used Vectorware. In another study, they used PyPro. So this is what I know. So these are usual transfection reagents that are used in the field. We have all there was the earlier study actually also used calcium phosphate, calcium chloride based transfection. So I think every transfection that you typically use for plasmids, you can just simply move over to minicircles as well.
Point. Third question, using minicircles seem to increase AAV vector purity. Any idea on exactly how this might be happening and why this is so significant?
Yeah, once again, very important question actually. So this is very important because you now see so many different AAV gene therapies getting approval. So this whole problem of backbone sequences in particles has now taken center stage, and rightly so, because these particles can actually elicit an uncontrolled immune response. When we now look at clinical trials and actually injecting these particles into patients at very high levels, so this is very crucial. So it is important to note that when you supply these backbone sequences as a starting material in your plasmid, they can actually start gaining the proximity to the hairpin structures like ITRs, and they can gain the ability to serve as templates, which will make further amplification of these backbone sequences possible. And there are literature reports that show that as the rep protein binds to the RB prime on the hairpin structure, these molecules with backbone sequences are prone to getting falsely packaged. And during this process, certain adjacent nucleotides to the RB prime motif can actually act as pseudo D sequences. So even if the D sequence is typically not present in these backbone genomes that are being amplified, certain adjacent nucleotides can act as pseudo D sequences, and this whole packaging can still happen. And that is corresponding to the observation what we have, that we see backbone sequences in these particles. So long story short, all of these suggested mechanisms point to the fact that by removing such sequences, such backbone sequences already from the starting molecules and making the starting molecules clean and precise, you can expect to end up with clean, precise, and therefore safe AAV vectors.
Very well, thank you. So now to the next and last question that we see here in the chat. You highlighted that ITR integrity is critical for AAV production. Can you briefly summarize your technologies that address this? So ITR protect and ITR rescue.
Yeah, that's correct. According to our, this was data from our collaborators, from our academic collaborators. From their observations was that during AAV genome amplification in the producer cells, you can actually expect truncated ITRs from plasmids to be corrected, but this process is neither reproducible nor reliable. Okay, and therefore, firstly, you have these genomes on your vectorized plasmid molecules, and they need to be rescued. This is a process called fold back replication. There is a nick at the TRS to begin with, and when you provide stably maintained wild type like ITR sequences in the starting molecules, I think we can free up the producer cell from these hurdles of having to correct these sequences. So you can actually expect to accelerate your genome amplification. And I think more importantly, since all of those ITR sequences are full and they are correctly present, all those secondary structures that are important, they can be formed accurately and precisely timed events can happen. And this will probably successfully help because it is similar to the AAV wild type virus, which has evolved to be efficient. So we can expect such similar accurate replication of these genomes. And you have a less chance for a read-through replication into the plasmid backbone. So that is the crucial aspect here. So this will explain why you see a reduced number of backbone sequences detected in your particles, even though you provide them in your transfer plasmid. Now, combining this idea with the Minicircle was the key advantage here. That was a key difference that we made. And this truly elevated the system to show the data that we saw shortly before, how we could significantly increase the levels of genomic purity. So if you take all of this together, I would say these new constructs that we now have at our disposal, they represent crucial strategies that need to be taken at the starting molecule level in order to combat this very serious issue of AAV vector heterogeneity, because this is real and this is coming, and this will have regulatory consequences down the road for all viral vector manufacturers.
Good, thank you very much. This was the last question, at least in the chat. Of course, everybody can always contact us via mail to pose further questions, and we are open to discuss, to answer the questions, to meet up if you're interested, of course, in a further collaborations. And at that point, I would say we can close the webinar. However, just one thing that I'd like to point out, please visit our website and follow us on LinkedIn if you can. Just look for PlasmidFactory GmbH and to get news and updates about our products and services. And feel free to join our fellowship. With that, I close, and I thank again Dr. Shankar for this interesting talk, and you, the audience, for registering and participating, and have a great day.
Thank you very much, everyone, for listening. Thank you, Dr. Stein, for this moderation, and thank you again for all the listeners, and wish you all a very nice day. Thank you.